We are just afternoon, so I think we're going to go ahead and get started. Thank you so much, everybody, for coming today to our November lunchtime lecture. This is actually our last lecture of the year, and it is part 2 of our staff symposium. So I think we're going to start with Dr. Manuela Del Forno.
Hello, everyone. Thank you for being here. I'm really excited to talk to you all about about one of the projects that I've been involved here at BRIT. So just to get started, you're gonna hear the word lichens a lot. So I just wanted to have a quick reminder what are lichens. They're a multi-species symbiosis. A symbiosis literally means living together. And in a lichen, what's living together is a mycobiome or a fungi, usually, formed by hyphae. And other examples of fungi are the mushrooms that we see outside. It's also composed of photosynthesizing organisms, such as green algae or cyanobacteria. We know other algae, for example, when we eat sushi or under the sea, but there are also a lot of terrestrial green algae. And lastly, there's also a community of microorganisms, such as bacteria, fungi, and other. And we also have a microbiome, for example, the one in our gut. So all of these coming together form what in nature we see as the lichen thallus. So even though it looks like a mini plant, a mini ecosystem, it is not a plant, but it is a mini ecosystem because we see all of these things living together. So it looks like a unique body. So today I'm going to tell you the story of the Lichen Team, and we're featuring today a little bit about the story of preserving the past, exploring the future, and some insights from the Philippines NSF project. So to start, I want to introduce you the heroes or the characters. So we have 5 in-country Filipinos working in this project. You met Yvonne, and we also have Erms and Cheska, who are arriving in about 2 weeks. They will also be joining us here at BRIT. And we also have Queenie and Arman. And the non-Filipinos are me, Manu, and then 2 other lichen specialists that work in specific groups, Bibi and Alisi. But we also have other people that have not gone to the field, but have then worked from far away. You've met Mam Paulina, and then we also have Alba here, who's helping us to identify certain species from the Philippines, and Adriano, who's also a specialist. And then we also have featuring Ken, who is one of the newest members of the Lichen Team. He's an undergrad in the Philippines. And then we also wanted to say that now all of this data that we've collected, they're all going to be online through the Lichen portal with the help of our staff here. So it's going to be accessible in these data portals and available to all researchers in the world. So the Lichens team started as a part of the National Science Foundation grant that we have. We started in 2018. It just ended a couple months ago. And if you want to hear more about it, we have a lot more details in my website. But this is the official name, Plant and Lichen Discovery in the Southern Philippines, and is led by the VP of Research, Dr. Peter Fritsch, who also has a talk right after this. And these are just some images of our fieldwork there. And the goals of the project, we have short goals and short-term goals, long-term goals. I'm not going to go through in detail, but I will highlight a couple of them moving forward. And I just wanted to share you about the Lichen Team rules. We're a very strong team, but we have rules. And the first one is safety first, because you know how the field can be very dangerous. We collect everything, meaning everything, not just the pretty lichens. We want to have an idea of everything that's there. When in doubt, pick it out. That's a very good rule, because you've all been in the field, or most of you have been in the field, and you're like, should I collect? Should I not? Is it common? Is it a weed? But when in doubt, you should collect, because you never know. And I can guarantee you have a lot of stories of a last specimen that I collected in the first expedition that I was part. And I thought, is this lichenized? Is this just a fungi? No offense to just fungi, but and it turned out that I said, it could be something interesting, and I collected, and it's actually a new species that you see in future slides. We are also respectful of everything, so colleagues, local communities, cultures, but also to the organisms. We don't just start, hurting other things. We're careful where we step and so forth. So be respectful to all living things and cultures. And then last of all, have fun. And what we hear about our team is that we work in the perfect taxonomic group, and we know it because lichens are not seasonal. So you can go any time of the year. They are also very photogenic. We take great pictures because they don't move a lot and so forth. So I just wanted to let you know that I could stay here all day talking about how many great qualities lichens have. But we do have a monster amongst us, and we all face this every day, which is the biodiversity crisis. And the tropical regions are the most susceptible for many reasons. And we know about climate change, and then especially in these habitats in the tropics, about habitat destruction and deforestation for many reasons. And we have experienced this at firsthand. So our objectives, shortened from that big slide with the many objectives, the Lichen Team is interested in exploring the diversity of the southern and describe all of the new species, publish new records, and so forth that we encounter. So this is taking place in the Philippines. And when you imagine the Philippines, you always imagine these beautiful, pristine places. But we don't go there to be in this beautiful lake. We go there to collect the lichens that are around this beautiful lake. So you see the lake here, but we are looking for the lichens. And some people might not know that lichens can also be around water. So here are some of these species that are found underwater or just around the border of these areas. And good for us, it's also available just on campus. So when we are in between trips, you can also just go collecting on the campus. We collect lichens that are growing everywhere, and these are some specific terms that people often ask, but they can pretty much grow anywhere, including in man-made substrates. We collect everything. And there's even some living organisms that may have lichens. And these, for example, in this walking stick from the Philippines, we saw something that looks exactly like another lichen here. Just have to go back. There we go. And so it's amazing how some of these have developed mimicry for some of these lichen species. And then how we do that, we collect a lot of samples, and you can see here, even sometimes we have extra tents. Just for the lichens, we also hold some of them when we run out of space. We collect, we also pick small fragments for DNA. And sometimes we have our presses and they sleep on our tents with us. So it's not uncommon that things are gonna start coming out of this while you're sleeping. But better that the lichens are protected than an animal coming at night and peeing on it, for example. Okay, we might need to go back. There you go. All right, so just a little background. There are about 20,000 of lichen species described for the world, and about 1,200 of these are already described in the Philippines. And this is a checklist that came out and had the most updated numbers that we use as a foundation. Oops. It's all right. Okay, let's go quick. Mhm. So this is the checklist, and from the total, we know that over 50% of the known families of lichens are present in the Philippines, and about a quarter of them. And this 6.5% of the species, we know for sure that we're gonna increase this number when we finish working with this group. There you go. So we just wanted to give a quick run of the numbers. We collected about 11,000 specimens of literally everything, something really big that could be like this, and something really small that is less than a centimeter. So we try to collect everything. And we've collected over 40 families, so we still have ways to go, but we are building these numbers up. We've offered workshops, we've had several lectures, we trained a lot of people, and we've visited a lot of communities. So we've had 6 expeditions, and as I said, a total of 11,000 and nearly 25 total, considering the duplicates that we will have. And we also have research. One second. It clicked. Hmm? Yes. So if I had more time, I would definitely know. There you go. So a little bit of our ongoing research, and I won't go into details, but my specialty is basidiolichens, and we've sequenced over about 300 specimens to have 200 to go, but we can already see here, all of these still are specimens from the Philippines. So we will have a lot of different new species to describe. We also have been working with this group, Lepidostromatales, that have a very unique ecology. They grow on the roadside banks, and definitely the community, when they see us looking at this, that they pass every day, have no idea of the value of the diversity of the lichens growing in this. And this is actually the new species that I told you that we collected in the when in doubt, pack it out comment. And this is yet another species that we found. And this is a new record for the southern part of the Philippines. I'm not going to go into detail, but this is just to explain a little bit of the research that we do. We take an integrative taxonomy approach. Which basically means we gather all the information available to come up with a species concept for each one of these lichen groups. And I'm working on the basidiolichens, but several other colleagues are working on other things. So I just wanted to also show you a video of our expedition, and then after that, I'll finish. And this is just on the latest expedition. This is a normal day in the field. It starts with a sunny day, if we're lucky, and then it ends in rain. But there you go, there's the rain. Some crustose lichens, some big showy lichens we collect on rocks. That one was a species that has a lot of ants underneath. Some bryophytes are always around collecting. Some free climbing. And this is now the processing of the lichens. We collect all of them, as I show a couple pictures before. We either press some lichens like a plant, or we let them dry naturally. Every night we do this for many hours. All the teams do, that's not just us. And this is now going to another island. We had to take a boat, and this is just a little bit of our baggage. Yes, right. Many dozens. Safety first. This is a very unique habitat in the Philippines. Is known as the bonsai forest. It's only occurring in a few islands, and this is one of them, the Nagat Islands. We also take many pictures, only pictures of animals. And a terrestrial crab, if you've seen it inside the plants. So this is the bonsai forest with a lot of these patches then of deforestation. There's heavy mining there. So all of this island is in danger. So that's why we return there. Processing even at night in our tents. The final number is 1,851. That's for expedition 6. We collected 1,851. But it's sponsored. All right, so a video talks a lot more than I could possibly try to explain to you what one of these. I just want to finish by thanking and acknowledging our colleagues. Some of them you've met, and there are also training some of these students. We also have students working in their masters now. Each one of them is working on a different group. Ermeline, who's coming, she's working with pyrenolichens.
Is everyone here interested in lichens?
So this is Sir Arman. He works in the education department, but also joins us. And then you've met Yvonne. She's working with this group of Graphidaceae in her PhD. Dr. Bibiana Moncada, she's the curator in the Berlin Botanic Garden, and she works with these big, beautiful lobarioid Piltigeraceae lichens. Alice, who visited last year, she's working with Usnea. And for Usnea, because this is so showy, we were able to get some common names for these in the local languages. And Francesca, who's also coming, working with these other types of lichens. And finalize by thanking everybody that has been involved with this project. Has been nothing but wonderful memories and a lot of new knowledge to share with the world. Thank you so much.
Thank you, Manu. We don't have time for questions, so we're going to move right on to Peter Fritsch talking about his research with blueberries.
Okay, well, that's going to be pretty hard to follow. Should have had her going. Hopefully we'll have time for questions from Manu later on. I'll try to be super quick. All right, well, I wanted to update you all on a project I've been doing with colleagues at Duke University and University of North Carolina. Sorry, NC State. Apologies for this is being recorded, right? Is this going to work okay? The slides? Okay. On the systematics and evolution of the true blueberries, Vaccinium section, Cyanococcus, and the Heath family, Ericaceae. And what's a true blueberry? I'll explain that in a bit. As soon as I get to, there we go. Okay. So who doesn't love blueberries, right? Super fruit, high in phytonutrients and dietary fiber. They taste great. They're one of the few major food crops indigenous to North America, interestingly, and one of the newest of the cultivated crops. There hasn't been a ton of work done on it. So 1912 was when the first plants were taken from the wild. And it's the star of a multi-billion-dollar agri industry. Okay, what is a true blueberry as opposed to a not a true one? That occurs in this section, Cyanococcus. Well, they're mainly Eastern North American. And there's two characters that you can always use to tell a true blueberry apart from any other. And they're both vegetative characters, interestingly. They're not flowering characters. The first one is this stripy bark. So it's, we call this eruptive periderm. So if you see a blueberry with that, chances are you've got one of these. And then the other one is even more reliable. This is a second year stem. And on the second year and third year stems, you can see these little dots all over the stems. And those dots are actually what we call stomata. Stomata are little holes that allow for air transport. Normally they're in the leaves, but these plants have them also in the stems. And so if you've got these little dots on the stems, you've got yourself a true blueberry. Yeah, so there's other characters in combination now too. But in any case, so the taxonomy of the group has been very unstable, I'd say. So the first one to look at this comprehensively is W.H. Camp. He was a curator at the New York Botanical Garden and contributor to the evolutionary synthesis of the 20th century. And he was one of the first to use ploidy in taxonomy to propose a model for the evolution of polyploidy. So what's ploidy? Ploidy is simply whole genome doubling. Just remember that. Plants do this a lot, and it's very important in their evolution to undergo doubling of their chromosomes and tripling and so forth. And so he developed a model of 25 species of diploids, tetraploids, and hexaploids, so 2, 4, and 6. And so going from, diploids to these tetraploids. So, it's a one-way street. Theoretically, it's not really likely to go back. And so you go from one to the other at higher level. And so the ones with the straight lines, that's called autopolyploidy. That's one species to another. And then the ones with the cross lines, those are merging, and those are two parents giving rise to a different species at a different ploidy level. So he had these very complex scenarios sometimes for tetraploids and hexaploids, and left it at that. Then came along this fellow Sam van der Kloet, Acadia University in Nova Scotia, Canada. And he didn't like Camp's treatment at all, and he lumped a whole bunch of his species together. So he ended up with only 9 species and divided them into like low bush types and high bush types. And so less than a meter tall is, you got this category low bush. You might have heard these terms, high bush southern, high bush blueberry, things like that. High bush 1 meter tall. And so the rest have been lumped, and all the high bush ones have been lumped into corymbosum. And you can see that 2x, 4x, 6x, that means different cytotypes. That's different ploidy levels within a species. And so quite different. He thought of corymbosum as something called a compilo species, which means it's a genetically aggressive species that takes the heredities of other species around it and incorporates it into its own genome, which sounds very like a horror movie type thing. But there's controversy around that topic, and it's not really clear whether something like that really exists. But there you have it, because this is the standard treatment now for the blueberry group. And it's the one in Flora, North America. So everybody's following this. And so when they see things that don't conform to this, they get really confused. And the blueberry breeders are confused by the names as well. So we decided we wanted to do some work on this to try to resolve this problem. And so we got NSF money for an investigation into the evolution and taxonomy of the true blueberries. We want to know how many species there are and what their limits are. And can stomatal characters in those stem characters that I mentioned help to determine ploidy? And what are the phylogenetic relationships? And was Camp correct, or was van der Kloet correct, or is it somewhere like in between? And so we've done a review paper already. This is in the journal Rhodora, just came out this year. And so it just reviews the whole topic in detail. Some of the methods, just real overview, field sampling of hundreds of plants in many populations throughout the range of the section. We examined thousands of herbarium specimens from US Herbaria, and that of course includes our own herbarium. We have a fantastic collection of this group in the Brit Herbarium. Ploidy level estimation through flow cytometry. I won't get into that, but it's just a technique to determine what the ploidy level is for over 500 individuals, and that's a lot for that technique. And high throughput DNA sequencing with the angiosperms 353 probe set. That just means we got gobs and gobs of DNA that we could throw at the problem. We finally incorporated all the key elements of modern monography. That's like an overview of a whole group of organisms, in this case, these plants. All right, so just right, cut to the chase. We basically disintegrated the high bush blueberry as it's currently understood. That means in van der Kloet's mind. And so we pulled out a bunch of species. All these were named by Camp, so homage to Camp. And some of them are diploids, hexaploids, and tetraploids. And I'll just give you an example because I don't have time to go through them all. I'd love to, but here's one, Vaccinium ascii. So it's a hexaploid, and it only occurs, it's very strictly endemic to mainly the East Gulf coastal plain with a couple of outlying populations. And this is actually pretty distinctive. The first time I saw it, I didn't know what I was looking at because it was buried in synonymy. It was, corymbosum. But gradually realized that this is distinct. It has this interesting blue-green vegetation, and it's got little stipitate glands on the backs of leaves and things like this. But what's even maybe just as interesting was our discovery of this. And Bob, you were with us when we went to this place. The first blueberry ever collected in Texas was this one. And it's actually, it's an escape from cultivation. Yeah, it looks like ascii, and it's been called ascii, and it's been called other things, but it's been the whole thing has been very confused. And you can almost always tell these escapes, and they occur in Texas, they occur in Virginia, they occur all over the place, but they're not ascii. They were considered to be ascii, but they're not ascii. Ascii is just very strict because these have glabrous leaves. They don't have hairs on the backs of the leaves like ascii always does. And you can tell them most of the time. And these have been crossbred with other species. So it's not really any wild species anymore. It's a cultivar. It's a cultigen that's escaped. So if you see one of these things, I don't know, if it's at a native area, we have to think about, like, do we want to keep it there? It doesn't really belong there. Okay. So the rest of the blueberries were called residual high bush species. And that's at the diploid and the tetraploid level. So these are all the plants that are left over after we pulled everything out. And these plants are considered to be the progenitor for high bush cultivars that we see, other than the one I just showed, which is called, I didn't mention, the rabbit eye blueberry. And these are all large shrubs with large falcate vegetative buds, often greater than 3 millimeters long. And then the rest of the variation is just all over the board. You can have high variation in like the leaf margin, the fruit color, the glaucousness of the fruit, hairiness of the leaves and the stems, et cetera, et cetera. And all that has been used to split this thing apart into a bunch of species. And really all of it is what we'd say taxonomically trivial. That doesn't mean anything for species concept. And really when you boil it all down, it's just two ploidy levels in this group. They look very, very similar. And the only way you can tell them apart is by characters that are actually associated with ploidy. Ploidy is thought to affect corolla size, larger corollas, larger fruit, and especially stomatal size and density. And these are the distributions of our two. You see there's got mainly distinctive distributions, pretty interesting, but there's this overlap in the Atlantic coastal plain and the Piedmont. Okay, so I want to focus a little bit, just a little bit on this stomata study, because this is my master's student, Eddie Esparza, and his graduate advisor, Matt Hale, and we all work together on the committee at TCU. So we're graduate associate faculty at TCU as well. And so just a little bit about leaf stomata. So you can see these, this is there's a hole in the leaf, basically, to let gas in and out and water. And so around the hole, you've got guard cells, a pair of guard cells, and they move back and forth to let air in and out. And then you've got subsidiary cells. And so that's basically it. And so he measured 20,000 of these basically for this study. Just a phenomenal job, really, especially for a master's degree. So I'll just cut again, not bore you with the details of the methods, but just tell you what we found. And we found a beautiful correlation between the stomatal size, that is the width, the length, and then also the density and ploidy. So you can see the blue is diploid, the green is tetraploid, and the red is hexaploid. And you can see the pattern. It's pretty obvious, but it's statistically significant. We ran it through statistical tests. And here's the density, and the density goes down, interesting, rather than up for polyploid. So you've got fewer stomata on the leaves, but still it's a beautiful pattern. And the nicest thing about this, I think, is to bring it back to the stem. Remember I told you about the stomata on the stem. Well, it turns out you can actually see these stomata on the stem. You have to have a high-powered microscope to see them on the leaves. You can sort of see them here with a dissecting scope. But you can see here, much smaller and they're denser. They're much smaller and they're denser in the diploids than they are in the tetraploids. So you can see this pretty much with the naked eye. So you can go out in the field and these things grow together. So you can go around and pick out the diploids and the tetraploids pretty easily. And that's, yeah, you can't really do that on a lot of plants. I mean, it's just luck that we got this stem stomatal character, really. And we were sort of lucky that we kind of stumbled on it as a good character to use. But that's called what? Serendipity. Yeah, dippity-doo-dah. All right, initial molecular results with polyploids added. So now we're going on with a little bit of phylogenomics. Getting a little technical here, so I'll just kind of skim over this real fast. We got 4 big clades: residual highbush that I mentioned, Pallidum, Eleatii simulatum, got a southern stipitate glandular clade, and Appalachian and northern clade. And so that's pretty much what we incorporated all the data together into one analysis for this. But we also wanted to kind of tease apart, see if we can figure out the origins of some of these polyploids. And so we use this method here and plus this method called structure, and that assesses admixture for genotypes in populations. So we got, for example, the origin of Vaccinium simulatum. It's a tetraploid, so where did it come from? Camp hypothesized an autopolyploid origin from V. pallidum, which is the diploid. So direct line evolution from one species to another based on chromosome doubling. And indeed, we get simulatum as sister to pallidum in the phylogenetic analysis. So that's really cool. And we get those high numbers there, indicate strong statistical support. And with the structure, this is structure. And so we're kind of using structure in a little bit of I don't know, unique way or a different way, in that we're looking at these K partitions. These Ks indicate the subdivisions of populations. And we don't have a lot of numbers for our populations. We might be cheating a little bit, but for the point of this, you can just see that you've got the orange and the square around it, that's simulatum. And so you've got the orange in pallidum, you've got the orange in Eleatii, and the orange in simulatum for K equals 4. And then they start to diverge as you subdivide the populations more and more and more, until at the end of it, you've got a unique simulatum profile with its own color system, unique pallidum, and then unique simulatum. This sort of tells us, sort of in a way, what it's telling us in the phylogenomic analysis, in that these things are closely related. Simulatum has diverged from pallidum, but we would interpret this as a autopolyploid situation, or at least it's consistent with an autopolyploid situation relative to an allopolyploid. If you had an allopolyploid, you would expect colors to combine in simulatum from other species. Okay, so just to summarize, our taxonomic revision entails what we call lumping of CALM species and splitting of vanderclots, so kind of right in between both of those treatments. Species identification has been greatly aided by the stem-stem model character, the size and the density. Autopolyploid, I didn't show you, but other examples are also supporting the autopolyploid rule in the section, and that's supported by this thing called tetrasomic inheritance, which I don't have time to go into in gory detail, but love to talk with you about it. So the next steps, more field work. I want to do, we're on a second year no-cost extension for this grant, and we have a little bit of money left over. So field work in Canada to check out the Canadian species, which we haven't done yet, and to go back, we've done field work in Florida twice now. We want to go back because there's some really weird things happening in Florida. No surprise, right? Oh, no, apologies to Floridians. Whole genome sequencing on selected samples, publication of species taxonomy papers toward a complete taxonomic revision of the section, careful study on stems to model characters correlated with ploidy, and then the documentation of hybrids, which nobody has actually ever done in this group, which is kind of hard to believe, but no one's done it with molecular data. Okay, that's all I've got.
I think we have time for one quick question, if anyone has a quick question.
No, this is fascinating, Peter. I have so many questions, but so I don't understand why you want to look at the F1 hybrids if most of the hybrids that you are finding are autopolyploids. Like,
Yeah, I didn't mention that there's can't believe there were hybrids, F1s, et cetera, introgressions everywhere. That was part of the problem with this taxonomy, is that it's all complicated by polyploidy, segmental polyploidization, and hybridization. And even Vanderkloot considered hybridization to be very important in the group. And so, you know, I didn't mention that before, but that makes it very much more complicated. We don't see that level of hybridization in the group just doing the field work. I definitely see hybrids or things that I think are hybrids, but they're not as prevalent as they would think. But I haven't been able to test it yet with molecular data. It's kind of been something we've been really wanting to do, and we've got genomic data in. I think we actually have it out now, and so we'll maybe be able to see what the results are for that. But we would expect combinations, you know, of characters and long branches, things like that for these F1 hybrids, and I'm hoping we're going to be able to see that. But so it's a little different than the polyploid situation. And in fact, there's a strong triploid block in this group. That means that you can't get a diploid crossing with a tetraploid very easily. They just won't cross. And all the blueberry breeders know this. And you know, you're only going to get crosses from within the same ploidy level. So diploids cross with diploids, tetraploids cross with tetraploids, and hexaploids cross with hexaploids. Yeah, and so sometimes they artificially induce tetraploidy from diploidy by using this chemical called colchicine, and they double the chromosomes artifactually. Then they can produce all these crosses and produce, you know, what they want to try to get to, which is ultimately a better blueberry.
Thank you, Peter. We'll move on to our next presentation from Tiana Rehman, Ashley Bales, and Jessica Lane.
Thank you, Jessica. All right. Well, I asked Erin's permission for our section to go last. You'll be hearing from 3 of us, but especially because today's lecture is a celebration of sorts. And if you're paying attention, you will hear what exactly we're celebrating. And in case we forget to tell you, at the end, we have some goodies to celebrate this thing that we will be telling you about. I haven't whetted your appetite yet. So many of you know this is a journey, it's a continuing journey, but you all have been a part of it. And it is specifically this aspect of digitizing our holdings. So today we're going over 4 sections: the role of the herbarium, specimen digitization as an opportunity, enhancing local and global collaboration, and then kind of future directions. So with that, I'll start with the first few slides. The role of the herbarium, I'm not going to belabor this point because many of you are here, support the collection already, work in the collection already. But this is a primary source of information for the study of biodiversity and environments over time, right? We don't have a time machine to go back in time, but our specimens represent that in some way because they were collected at a past point in time and place, perhaps, responding to the environments of that time and place. So just some examples of the uses of these are species discovery and classification, looking at evolutionary histories, so on and so forth, defining conservation priorities. But ultimately, our obligations as custodians of these specimens that you all help us accomplish is to safeguard these, to safeguard them and to make them accessible, which is why this priority that the herbarium has had to digitize our holdings is really what we have spent many years concentrating on and will concentrate on into the future. And just encouraging you, there's a wonderful article. Some of you recall there was an article put together by Vicky Funk. Oh, I don't even remember when, 2002, 100 uses for the herbarium, or at least 72. This is an update of sorts. It's not a list, but this particular article is very easy to read, and it kind of talks about collections-based research, which is one of our priorities here at the garden. So read more of that if you'd like. But this slide also now shows you that this is a global effort. So you are looking at statistics from Index Herbariorum, which is hosted by the New York Botanical Garden on the left. And so this is from their 2022 report. There are 3,567 active herbaria around the world, representing 396 million specimens in 183 countries with over 13,000 associated staff, some of whom are in this building right now. You can see that map showing the density, the darker blue representing higher concentrations of collections. On the right-hand side, you're seeing some information about the top 10 largest collections in the United States. So to set the scope of the problem, or the scope of the opportunity, I should say, you'll see that we're 9th in that list. So that has us listed at 1.48 million specimens in our holdings today. Sorry, I think I broke it. There we go. So many of you were standing in this building today. Some of us are on Zoom, but those of us that are in this building have the opportunity to walk over to the herbarium and look at the cabinets directly. So these are preserved in cabinets. That's a microclimate within a larger climate-controlled building that allows us to preserve these specimens. But it does make it inaccessible for any kind of work that we'd like to do, as you've seen Manu and Peter both talk about already. So having an understanding of this by digitizing them, and you'll hear what exactly that means in the following slides from Jessica, allows us to better understand what we've got and how we can grow and how we should grow and where we can prioritize growth. There was an important consensus report that you can see on the right side of the slide right now that came out, a few years ago, and it was talking about the importance of these biological collections for addressing some of the major problems we have today, and the importance for research and education. Specifically, digitization is a method to guarantee access to all these objects, to allow them to be searchable so we can prioritize research into some of these specimens. We can't do it to all of them. A way to preserve them, because digitization allows people from all over the world to see the specimens and prioritize which they'd like to physically request. Interoperability, the idea that you can look at these with new lenses if those data are liberated in a way that allows you to use these tools. And then, of course, interaction, a way for all of us to interact with these specimens. So setting that briefly as the stage for the value of digitization, I'm going to hand this over to Jessica, who will tell you a little more.
Thank you, Tiana. I'm going to talk a little bit about the process of digitization and where we stand in our collection. First up is imaging. So the goal with imaging is to have every specimen represented by a high quality image that we can put online. This is one of our biggest projects in terms of numbers. Last year, we imaged 160,000. This year so far, we're at 150,000. The big question is how much of our collection is imaged. I'm going to put that question on hold. I'll come back to it in a second. First, I want to talk about Google Arts and Culture. You might have heard of this initiative for working with art museums, cultural museums to put images of art and cultural artifacts online. They also work with natural history museums. We started our project with them in April 2021. They loaned us equipment and they granted us funding to hire a contractor, Jared Stone, to image specimens full time. We started with the theme of trees. We imaged a lot of tree specimens. We ran out of tree specimens to image, and so we started imaging the whole collection. Since April 2021, we've generated 450,000 images with this project, which brings us to 100% complete imaging for our herbarium sheets. Pause for cheering. Of course, this comes with the caveat, we still have backlog material, unprocessed material, non-vascular collections that need to be imaged, as well as keeping up with our new acquisitions. So there's still lots of imaging to do. Now that we have all of these images, we need to transcribe the labels so we have data in our database that we can search for these images. Traditionally, this was a pretty boring process, sitting down, reading the label, and typing all of that information into a database. We have some modern tools like optical character recognition and artificial intelligence that let us use machines to make this process go a little bit faster. As an interim goal, we're hoping to have all of our records searchable by broad taxonomic and geographic categories. We call that skeletal transcription. Right now, we're sitting at 81% skeletally transcribed and 30% fully transcribed. And last up is georeferencing. The goal is to have all specimens associated with geocoordinates and uncertainty measurements so that we can see all of our specimens as points on a map. This is a pretty new project for us. We only started georeferencing in earnest about a year ago. And so far this year, we've done 41,000 georeferenced specimens. That's about 8% of our collection. For transcription and georeferencing, our focus is really on targeted projects that Ashley is going to talk about next.
Thank you, Jessica. So you guys heard from Manu and Peter about some of their projects that involve collecting specimens, and those, being collected today, they have GPS units. Those specimens are born digital, but for everything that exists, hundreds of years ago until now, we're focused on two current grants, our All Asia grant and our Texas, Oklahoma, our Torch grant. So for All Asia, we have over 50,000 specimens across the continent, and more than half of those are collected from the Philippines. So even though we have our Southern Philippines grant right now, back in the 90s, we had other projects called the Philippine Plant Inventory. So we have quite a lot of specimens from the Philippines. And we have just updated last week. Thank you, Craig Meyer. We are almost done with the transcription part. So most everything has been imaged at this point, and so we're working on the transcription portion and eventually the georeferencing. So there was supposed to be a picture of Craig, but he's back here. But this is Craig's wonderful graph. He has been working a lot with what Jessica mentioned, machine learning, AI, to get this large amount of specimens transcribed. And so as of last week, we are less than 9,000 specimens to transcribe remaining, which is a huge accomplishment. So just, of the other countries in Asia, some of the unique specimens that we have that we discover through this digitization process, on the far left is a specimen from the SMU collection that was from North Korea. It's our only specimen from North Korea. In the middle is from the NLU collection from the small country of Cyprus. It's an island country. And then on the far right is from the Vanderbilt collection, mid-1800s from India. And then on to the Torch project. So Torch, we've been working on for a little bit longer, started in about 2019, I believe. And from Texas and Oklahoma, we have nearly 230,000 specimens, so quite a lot. And we are completely through with the imaging and transcription phases, although we're still receiving more specimens through gifts and exchange from other herbaria, but we're about 30% through with georeferencing. And like Jessica said, we've been doing that for about a year now. And just in the two specimens that are featured, one of the oldest specimens from Oklahoma that we had from the mid 1800s. And then through the georeferencing process, our staff and volunteers have found many ghost towns in Texas, which have been really interesting. Towns that no longer exist are now under a lake, something like that. And then, let's see. There is one more slide for you, I promise. The next slide, I kind of go more into georeferencing. Ah, there we go. Here we go. So all of the dark green represent counties that have been completely georeferenced. So we are so close to 100 counties in Texas that have been completely georeferenced of just our collections. Does anyone know how many counties total are in Texas? Yes, correct. We have the most counties than any state. So we have been taking this county by county approach. And so you can kind of see the pattern. We focused really on North Central Texas. Dallas is our last county just within, the DFW area. And we're going into the Panhandle range. And then afterwards, we're going to use more collaboration with other herbaria in Texas and Oklahoma to tackle the rest of the state. And where does this data end up? Y'all have probably heard us talk about our Torch portal. So we put all of our data there. That's where you can search all of our collections. And this data is aggregated into other places like iDigBio and GBIF. Future goals, Tiana, to end us. And I think we got time.
We do. All right, so this is just to wrap us up briefly, but so you've heard kind of some of the successes. So you heard that we're at 100% imaged for our filed out sheet collection, which is spectacular. And 81% of that is skeletally transcribed, so we can access it. But digitizing these holdings has really set us up for kind of the next step and the growth and infrastructure changes we'll have to make to these specimens. We have a lot of specimens here. The building can handle more, but we're getting close to having to figure out how to best take care of these specimens. So digitizing these means no matter what state, if we're carting them in a cart from the upstairs herbarium to the downstairs herbarium, they'll physically still be, or they'll digitally still be accessible. But lastly, this is a graphic that many people have shared in many different presentations. And the whole idea behind this graphic is that it originates with a physical specimen. That is at the nexus down there in the bottom, you can see. And then there are various extensions to this. So the first showing in there, digital specimen records, data about the specimens. So those are the transcriptions. Then you've got field notes, field images, morphometric data. You can see where Manu's presentation of working with specimens and Peter's presentation also fit into this idea, but this is all built around that nucleus of these surrogates that are placed online that allow people to cite a physical and a digital specimen. And so that's really the next step of where we're planning on looking at how these specimens and what we can do as custodians to make them accessible and useful. With that, just a big thank you to our volunteers that have participated in every aspect of this, our virtual volunteers who are not with us, our armchair botanists, National Science Foundation, Google Arts and Culture, Mr. Patrick Newman, our executive director in the Fort Worth Botanic Garden, and of course, colleagues and volunteers, yes, I said it again, and supporters. On the right-hand side, you can see some of the initiatives here at the garden. So we've actually been with our living collection staff sampling our living woody plants. That was a volunteer class, and they collected, I think we collected maybe 12 different woody plant species that day, and then they're being mounted on the left-hand side of that screen. So you may see yourself in there. So with that, the three of us, thank you for attending and thank you for participating. So cheers.
Thank you. Are there any questions for Tiana, Ashley, myself, or for any of our speakers?
The blueberries we heard about edible.
Are the blueberries edible?
Everything's edible once, according to Bob. Yeah, they are. Yeah, and in fact, all of those, many of the species in the whole section have been used to improve our cultivated crop of blueberries. Oh, yeah, these are primo. Yeah, a lot of them. Yeah. And in fact, the whole tribe, I don't know if there might be some, but the tribe is like 2,000 species. And most of them are just like that. They're blueberries. Some of them, one in the Philippines is just, it tastes like grape jelly. Yeah. Not been touched for cultivation yet.
Yeah, I love this set of talks. It's just wonderful to hear about what everybody's doing. And I have one question for Tiana and colleagues about the herbarium. I can see that you have the extended specimen, and I wonder how easy or difficult you think is to get funding now to finish the parts that we need to finish in terms of georeferencing mostly, and maybe digitizing the rest. And maybe Jesse or Ashley can.
Tiana was busy cutting cookie cake.
That is an excellent question. And I think that's something that I didn't get a chance to wrap up at the end that you noticed as we were giving the presentation. You know, we have 1.59 million specimens. And of the ones that are targeted, Ashley presented what, 50,000 approximately for the Asia grant, and then 240,000 or some odd for the Torch grant. We have had small piecemeals that fuel getting those completely digitized. So it's a whole nother strategy to figure out how are we going to find the funding to complete the digitization of the remaining specimens in there. We have been very fortunate to find kind of unique and distinct partners like Google Arts and Culture that have allowed us to image 450,000 specimens. I mean, that's rather unheard of. So that was pretty phenomenal collaboration with them. And hopefully in the new year, in the first quarter, you'll see some tangible results of that collaboration. So stay posted for any other announcements from the Herbarium. But I think it will take rather innovative looks at things. And you yourself have been talking about some great examples of how we might complete that and what we could do to prioritize some of those specimens. But, you know, our primary funder for the digitization of the collection, aside from Google Arts and Culture, has been the National Science Foundation. And a lot of those proposals were targeted around thematic collections networks, which you'll hear us say TCNs all the time. And that was the idea that we need to group these specimens into a package that has research applications directly for one sort of thing. And that unfortunately didn't allow us to do everything. And so I don't know, we're gonna have to be creative. And I think that's why it requires all of us, any of you that have ideas or places we can go or ideas to how we can market some of this, I think will be useful. Anyone else want? Yeah, go ahead.
Marketing just triggers the thought. So now that things are digitized, what volume do you expect of researchers coming in through the digital portals? And do you have the traffic and volume and ability to support this increased activity?
It's okay, we'll share. So thank you, Lee. That was a good question. So something that Ashley showed on one of the screens was where we're reporting our data. So we have them in the Torch portal. They also then eventually get reported out to the Global Biodiversity Information Facility. Researchers are daily, by the second, by the minute, downloading data sets of specimens and referencing those in works in their own research. And so in that way, we're handling that through those kind of global aggregators for where those specimens are going. But there is, you know, one of the biggest things about digitization is people not knowing where we stand. So we consider this part of our effort to make sure you all know kind of where we stand in our digitization journey. Because there's a lag between our database and when it gets up to the Global Biodiversity Information Facility. So that's one way we're handling that. And there are analytics at various stages to look at those things and look at traffic.
I think we have hit 1 o'clock, actually a little bit after. If you would like to stay for cake, and I'm sure our speakers would be happy to individually answer questions, but if anyone needs to get going, I want to set them free.